Transcription, the copying of genetic information from one strand of DNA into RNA, is termed as transcription. The principle of complementarity governs the process, except adenine forms base pair with uracil instead of thymine. In transcription, only a segment of DNA and only one of the strands is copied into RNA. Why it is so? Just listen. If both strands act as template, they would code for RNA molecule with different sequences, and in turn, if they code for proteins, the sequence of amino acids in the proteins would be different. Hope you understood. Now secondly, the two RNA molecules, if produced simultaneously, would be complementary to each other, hence would form a double-stranded RNA. This would prevent RNA from being translated into protein. These are two important points. You should note it down and learn well. Now, transcription unit in DNA is defined by three regions in the DNA, namely a promoter, the structural gene, and a terminator. Here you can see the three regions in the transcription unit. Try to understand the promoter, structural gene, and terminator region. Now, the promoter is a DNA sequence that provides binding site for RNA polymerase, and it is located towards Y' end of the structural gene. The terminator is located towards 3' end of the coding strand, and it defines the end of transcription. Now, the strand that has the polarity 3', 5' acts as template strand. The other strand that has the polarity 5', 3' acts as the coding strand. All the reference point while defining a transcription unit is made with the coding strand. Here you can see the template strand and coding strand. In transcription, only a segment of DNA and only one of the strands is copied into RNA. Now, the process of transcription include initiation, elongation, and termination. Now, transcription in prokaryotes, first let us discuss. There is a single DNA-dependent RNA polymerase enzyme that catalyzes transcription of all types of RNA in bacteria. So, there are differences between transcription in prokaryote and eukaryote. First, we are discussing about prokaryotes. So, in bacteria, there are three major types of RNAs: mRNA or messenger RNA, tRNA or transfer RNA, and rRNA or ribosomal RNA. mRNA provides the template, tRNA brings amino acids and reads the genetic code. rRNA plays structural and catalytic role during translation. At first, RNA polymerase enzyme binds to promoter and initiates transcription. This process is called initiation. Here you can see the RNA polymerase enzyme binds with the promoter region with the help of a factor, namely sigma factor or initiation factor. So, the RNA polymerase associates transiently with the initiation factor to initiate the transcription. The main enzyme required for transcription is DNA-dependent RNA polymerase. It also catalyzes the polymerization in only one direction, that is 5', 3'. It uses nucleoside triphosphates as substrate and polymerases in a template-dependent manner following the rule of complementarity that you know. Here observe this diagram and try to understand three different stages, namely initiation, elongation, and termination. The polymerization follows the rule of complementarity, and it also facilitates the opening of the helix and continuous elongation, the second step. Once the polymerase reaches the terminator region, the nascent RNA falls off, so also the RNA polymerase. This results in termination of transcription. Here you can see the RNA polymerase binds to the terminator region with the help of a factor, namely Rho factor. The three steps are shown here once again. Observe and listen. Try to understand. In bacteria, the mRNA does not require any processing to become active. That is one major difference. Then in bacteria, transcription and translation take place in the same compartment. In eukaryotes, there are two additional complexities in the process of transcription. That is the difference. Additional complexities we are going to discuss. First one, there are 3 RNA polymerase enzymes in the nucleus. The RNA polymerase 1 transcribes rRNA and RNA polymerase 2 transcribes precursor of mRNA, and RNA polymerase 3 is responsible for transcription of tRNA and snRNAs. The second complexity is that the primary transcript contain both the exons and introns and are non-functional, and hence it is subjected to different process, namely first one, splicing. It is a process where the introns are removed and exons are joined in a definite order. Here you can see the process in detail. The introns and exons are shown here. How the splicing is conducted. Just observe. In capping, an unusual nucleotide, that is methyl guanosine triphosphate, is added to the 5' end of hnRNA. Here you can see the capping. This nucleotide is added here. In tailing, adenylate residues are added at 3' end in a template independent manner. Just observe here, this process is also called polyadenylation. It is the fully processed hnRNA, now called mRNA, that is transported out of the nucleus for translation, that is for protein synthesis. Once again, the different steps of transcription in bacteria is shown here: initiation, elongation, and termination. This picture explains the process of transcription in eukaryotes. Hope you understood the process of transcription in prokaryotes and eukaryotes well.